I want to talk to you about a bit of my research, and I'm really interested in leaves, really. And I mean, you normally think of leaves and you think of things like this, these flat things that we heard about this morning with that lovely talk from Keiko. And she kind of linked this talk really nicely in a way in that she was talking about plants, these leaves, they have mouths, and they're basically the way that plants eat. Right? They eat the air, they eat carbon dioxide out of the air, and they build their bodies out of it. And this is what most leaves look like, and that's what most leaves do. But leaves have also evolved to make a whole range of different shapes and sizes and patterns. And one particular type of leaf that I've spent probably 7 years working on is these leaves here. And these are carnivorous plant leaves. Now, I know they don't look particularly like leaves, but each of these type of carnivorous plant trap evolved from a flat leaf, just like those ones in that picture that I just showed you. And they evolved from that into these very complex, cup-shaped, hollow structures. And each one of these is highly adapted to trap invertebrates. Basically, they eat tiny animals. So these two eat ants mostly, and this one eats small aquatic invertebrates. And you can actually see here, this is a little copiapod that's half stuck in the mouth, right? So all of these three types of leaf have evolved to feed the plant, but in a very different way from what we heard about this morning. So instead of taking carbon from the air to build the body that way, they extract nutrients from the bodies of dead animals inside them, essentially. And I'm really interested in morphogenesis, so how plants build their bodies, how they shape themselves, and in particular how that has evolved. So the question that I want to talk about today is how these three very complicated, kind of very different structures, how they evolved from a flat leaf, right? And when we think about making shapes in biology, we have to think about development, right? Now, and the really cool thing about all of these leaves and about flat leaves is that they all start off looking almost the same, right? So all plant organs, in fact, start off looking very, very similar. So if we look, these are the mature organs of three different leaves. This is an Arabidopsis thaliana plant, so this is one we heard about today, and I'm sure you'll hear more of. This is a Sarracenia. And then this is a utricularia, so an aquatic suction trap. I'll be talking a lot more about this one, because this is the one that I do a lot of my research on. But the point to take away from this is that all three of them grow from these small dome-shaped primordia. So this is an Arabidopsis primordia, Sarracenia, and a utricularia, and they all look the same. They're essentially kind of just shapeless bumps of cells, right? So the big question is, how do you go from making something which is just a dome of evenly shaped cells to making such different shapes? And how has the developmental program that goes from this to this flat leaf been modified to make this hollow, cup-shaped, complicated structure? And kind of building on that, each of those carnivorous plant traps that I showed you, each of them has evolved independently. So there's actually been at least 3 transitions from a flat leaf to a hollow trap. So it's happened multiple times. So to me, that shows, firstly, that it must be useful, and secondly, that it must actually not be that hard to do, right? Because it's managed to happen multiple times. So hopefully, I'll be able to talk you through today the mechanism by which we think this evolution has happened and the development that underlies that. But first, we need to talk a little bit about leaf development, right? Now this is a shoot apical meristem, a very zoomed-in version. So in the middle here, we've got these undifferentiated stem cells, and around the flanks here, we have primordia. And these are the things that grow up to be leaves, whether those are flat leaves or carnivorous trap. You can already see here that this leaf has started to flatten. And this is an Arabidopsis meristem, and this leaf will eventually become completely flat. And we know really well from many, many years of developmental genetics experiments that there are 2 classes of gene which really regulate this leaf flattening. And they divide the leaf into two. So they give it a blue half on the adaxial side, which is like the top surface of the leaf, the flat leaf when you think about it. And this orange half is the abaxial side, and that becomes the bottom of a leaf. And in each of these are different classes of genes expressed. And they kind of split the leaf in two, and we need the boundary between the two of these to make the leaf blade grow. And I'll show you what I mean by that in a moment. And this is just to show you that when you go from a primordia, like I showed you there, to the adult leaf, this blue and orange, adaxial and abaxial division is continued throughout the entire leaf. And that means that we have these two different, like broad different cell types. So we have these palisade mesophyll at the top and spongy mesophyll at the bottom. So we can see cellular characteristics that divide the leaf in half, essentially. But we know that we need these two different identities to make a leaf flat, because if we have a mutant where one of these two is taken away, then we don't get a leaf blade forming. So these are antirrhinum leaves, and it's like a classic developmental genetic system in plant science. And this is a mutant. It's called Fantastica. So hopefully you can see that this is very flat, whereas this one, it's almost needle-shaped. So it hasn't got any leaf blade forming. And the gene that's mutated in this is actually responsible for making the top half of the leaf, the adaxial identity. So this is what we call an abaxialized leaf, and it's completely radial. So we know that the interaction between these two surfaces of the leaf is necessary to make the leaf blade grow. For a long time, people kind of hypothesized that maybe this is somehow underlying different shapes or diversity of leaves, but we didn't really know how it happens. And that's partly because no one had managed to do any genetic experiments in carnivorous plants, and also because the mechanism for how that interaction between the top and bottom half of the leaf, how that makes a blade grow out, is pretty poorly understood, right? So, so I spent several years working with this plant here, and it's called Utricularia gibba. It's a carnivorous plant. You can see its little traps here. It's a bit of a strange plant because, as well as being carnivorous, it's also aquatic, so it floats in the water. And what you see here is fairly representative of the whole plant. It doesn't have any roots. It just kind of floats in the water and sucks in tiny invertebrates into its traps here. And the nice thing about Utricularia, I mean, this is a very zoomed in thing. A Utricularia trap is about 1 millimeter across, right? And that's one of the reasons why we use it. You know, like it's the Sarracenia trap or the Nepenthes trap is beautiful. Some of them can be about 15 centimeters long. But they're difficult to grow in the lab and kind of a bit laborious to shove under a microscope, right? So the really nice thing about Utricularia is that we can grow it very easily in the lab, and we can do a lot of genetic and developmental experiments on it that I'll talk through with a little bit. But first, just to show you how the trap works and what it looks like, really. So this, okay, this is a Utricularia trap. And it's a very slow-mo video. Can you see, this is a little copiapod swimming past, just to show you this. Swimming, like it kind of swims up here, touches one of these little hairs, and then gets very quickly sucked inside. Honestly, it's more impressive than when I say it. Sorry the videos aren't working. So yeah, it has this really cool suction mechanism, right, where it has this airtight seal around the door. This is the mouth with the door here. And essentially what it does is it's under, all this whole thing's underwater, and it pumps out water from the trap. So it forms like a partial vacuum inside. And what happens is this touches a little hair on the door buckles, and then the water rushing in sweeps the copiapod, the small invertebrate, inside and traps it there, and it gets digested. Right. And this was supposed to be a nice 3D video of showing you the 3D structure. But basically it has this very thin wall with an opening at this end. So almost like a cup shape, a hollow inside. Oh, it works. there we go. Yeah, okay. So, yeah. You see, you've got this very, very thin sheet. And essentially the sheet of the outside of this is essentially you imagine like a flat leaf that's been kind of bent around. And it's that bending, that sculpting of this cup from a flat sheet that we're interested in. So to understand how that happens and how it might have evolved from a flat leaf, we looked at the developmental process. And if we look from very early on, you can see that it starts off as this little bump and then gradually forms this sphere that then deforms to make a mature-looking trap with a hole here, this mouth that sucks in prey. But we need to really look early on to understand how this is working. So from this initial bump, you see this, you get this slight indentation in one side that then forms the inside of a trap. And what we really want to know is one of these sides the top or the bottom of the leaf, say? Like, how are these adaxial and abaxial genes working and coordinating to make this trap form? And we can look at where genes are expressed using various different methods, but we use this in situ hybridization, where essentially you can stain for where the RNA of a gene is localized, so essentially where that gene's turned on. And I've colored them here. In blue is the adaxial genes, and in yellow is the abaxial. So this is the top of the leaf and the bottom. And the top of the leaf is localized really purely on the inside of the trap, and the bottom on the outside of the trap, right? So that gives us this idea that maybe the top of the leaf evolved and curled round to make the inside of the trap. And we had some very early images of these things which looked like we had a very restricted domain of adaxial identity, so the top identity, right? And we wanted to test whether just restricting that blue region, the adaxial region, was enough to change the shape, or whether or did we need like extra changes? So did we need local changes in growth and patterning as well, right? So to do that, we did some computational modeling. And essentially, what we did was we had basically this system where, to simplify it slightly, essentially you have a boundary domain in green here between the adaxial and abaxial. And that boundary domain basically patterns the direction of growth, right? So everything is growing in parallel with that green domain. So whatever the shape of that green domain controls the direction of growth, right? And we express that mathematically using these polarity fields and controlling growth in 3 different dimensions, right? So, to cut a long story short, what we did was we started off with this even domain, like in a flat leaf, half and half. And when we grow, what you can see is you get a flat sheet getting made. So that's nice, that's what we know happens in a flat leaf. But what happens if we reduce this adaxial domain down? What happens then? We haven't changed any growth rules within each region. Everything's still the same. It's just still getting patterned by the boundary between these 2 regions. And when you do that, you can see that you get something like this hollow structure forming. It looks actually pretty similar to a young carnivorous trap. And we then tested this with some biological experiments to show that actually, yeah, if you remove one of these identities, you don't get traps forming, and if you remove them at different points, you get different sorts of deformed traps that fit with the model. But what I wanted the take-home to be from this really is that we can use kind of computational modeling and experiments in kind of unusual plants to really to understand or identify quite simple mechanisms that can underlie evolution, right? So the prediction from this is that actually the reason that you get carnivorous traps, these cup-shaped traps evolving multiple times, is because that it's such a simple shift, just a simple restriction of gene expression of one half of the leaf can easily make this cup-shaped form. So perhaps like it's an easy developmental route to take to evolve carnivory. And as a side effect, the developmental model that we made really helps us understand a bit better the mechanism for this patterning of how the flat leaf becomes flat. And we have lots of hypotheses from that we can test. Right? So yes, I'll leave it there. Just to say thank you to everyone who I worked with, because this work was work from a lot of people developing a very unusual plant to work with. Mostly these two people here, Beatrice and Jay, and Rob Bellow here, who did some of this work during his PhD. And this is my postdoctoral supervisor, who supervised the work, called Enrico Cohen, and he is being a classic, I would say, supportive PhD supervisor, preventing a PhD student overstretching himself. Yeah. So thank you all for listening.